Well, hello everybody. Welcome to this first video of the third unit. We're going to be talking a little bit today about the central dogma, but not too much because most of you seem to have a pretty good understanding of that material. And we'll spend the bulk of today's review video talking about different mutation types and a little cheat sheet that you'll be getting from me that'll serve as a good study guide for this week's material. But first, like I said, let's talk about the central dogma. And even before we talk about the central dogma, let's talk about DNA itself as a molecule, because there are two things that came up in some of the reflections that indicated some confusion here. So as a general molecule, DNA is double-stranded, so we have one strand of DNA doing this in the double helix, and its bases point inward, and we have the other strand of DNA doing that, and its bases point inward as well. And DNA is also anti-parallel, so this strand is going 5' to 3' in that direction. Whoop, let me get that 5 in there. Oh, now I lost the 3. So try to strike the balance. So that strand is going 5' to 3' in that direction. That means the other strand must be anti-parallel, and it must be going 3' to 5' in the same direction, so that we can have this base pairing work. Some of you were a little bit confused on this prime notation. Just to put it simply, there is always a phosphate group at the 5' end. That's what determines the 5' end as being the 5' end, and the 3' ends of DNA have a hydroxyl group. So chemically, that's how those ends get their name. If we want to be a little bit more precise in the naming, the reason why we have these prime names, here I'm drawing a ribose sugar. If it's DNA, we're going to have a hydroxyl group here, and we're going to have just a proton there. This is an oxygen here as well. And the base would be pointing inward, so whether it's adenine, cytosine, guanine, or thymine, that would be there. And then we have a phosphate group coming off here. Now, chemists love to number things. I make this point briefly in the lecture. And all of the carbons that are present in the base are numbered so that chemists can orient themselves to that. And ideally, all of the carbons in the ribose sugar should be numbered as well. Hopefully you know from chemistry that all of the vertices of these are actually carbons. So there's a carbon at each one of these dots that I've just drawn. And these need to be numbered as well. But then we'd have an issue, right? If we were talking about carbon number 4, we would be saying, well, is it carbon number 4 of the base or carbon number 4 of the ribose sugar? So in order to make sure that there's no ambiguity, all of the carbons in the base just get their number, and all of the carbons in the ribose sugar get their number and a prime. So this is the 1' carbon, the base comes off that, the 2' carbon has the hydrogen, the 3' carbon has the hydroxyl, 4' carbon here, and the 5' carbon is linked to the phosphate group. Well, if we have a phosphate group at the beginning of any DNA strand, what carbon is it linked to? The phosphate group is linked to the 5' carbon, that's why we call that the 5' end. And what carbon is the hydroxyl group linked to? The hydroxyl group that ends every strand, it's linked to the 3' carbon, that's why we call that the 3' end. So a little bit of clarity there, hopefully. The second thing to talk about real quickly on this is the semi-conservative replication. That seemed to give some of you trouble as well. Semi-conservative replication is the idea that when you're going to replicate DNA, the first thing you do is separate the two strands. So I'll try to draw that like this. So these are the bases pointing inward, and these are the, these are the bases pointing inwards, and these are these bases pointing inwards, just like we had before. And this is the 5' to 3' strand, and this is the 3' to 5' strand. So I've just pulled these two strands apart. And then when we do DNA replication, we use each of those old strands as a template for building a new one. So now the new strand is drawn in red, and we use the rules of base pairing for that. So if there's an adenine here, we put a thymine here. Cytosine here, we put a guanine here, and we do the same for this other one. And now we've gone from one DNA molecule to two identical DNA molecules, but what you'll note is that each of these two quote-unquote new molecules is actually composed of one old strand and one new strand. In other words, they are half conserved. We're keeping half of the old in each of these molecules here, so we say that's semi-conserved or semi-conservative replication. So in a nutshell, Semi-conservative replication is the idea that when we replicate DNA, we separate the two DNA strands, we keep an old strand in each new molecule, and we put a new strand in each as well. So anytime we replicate DNA, we actually are half old, half new, half old in the two molecules that we end up with. Now on to the central dogma. It seems a few of you just didn't quite get what we actually meant about the central dogma. The central dogma of molecular biology is meant to describe information flow. And that information in all biological systems flows from DNA to proteins. Proteins are the machines of the cell. Cells are alive because of the actions of proteins. Proteins do everything that sustains life. But they don't last very long. They need to be replenished. They need to be made constantly. And the instructions for making those proteins are found in DNA. So DNA holds the information. Proteins are the manifestation of that information. And we actually need a middle step to join these two, and that's where RNA comes in. So the central dogma states that information flows from DNA to RNA, ultimately becoming proteins. And in fact, the RNA that we're talking about here is typically messenger RNA, and it is a copy of the DNA gene, just in RNA form. So the process of copying a DNA gene into an RNA molecule is referred to as transcription. And the process of decoding that RNA molecule into a protein is referred to as translation. And I made the point before in the lecture that these words actually mean something because the language of DNA is nucleic acids and the language of RNA is nucleic acids. In fact, there's only a very small dialect or accent. RNA pronounces its T's as U's, is the joke I always like to make. I was Brooklyn raised for a very, very long time, and still when I get angry or excited, I lose my R's when I'm talking. And RNA is a version or a dialect or an accent of DNA, except the thymines are pronounced as uracil, but everything else is the same. So truly, we are copying or transcribing nucleic acids into nucleic acids. If we go from a recorded English report to a typed English report, that's transcription. When doctors have their notes transcribed, they're going from an audio version to a typed version. Same language, still English. But when we go from English to Spanish or Spanish to Chinese, we're translating, we're switching languages. And here we're switching from the language of nucleic acids to amino acids. So truly a translation. So hopefully you'll never confuse these terms because they mean something. Nucleic acid to amino acid is transcription to translation. Switching languages here, biologically speaking. So that's the central dogma in a nutshell. Now what that allows us to do is talk a little bit about how that process works. So if we just have a very, very simple sorry, I'm writing and saying something at the same time, which is not ever good for me. If we have a very, very simple code here in DNA, we know it's DNA because we have thymines present, and we want to first go through transcription. Remember, transcription is not going to change our language. So we just have to remember that accent. Well, adenine is pronounced the same way. RNA pronounces its thymines as uracil, so we'll change that. So this would be the end result of transcription. This is a DNA form of our sequence. This is an RNA form of our sequence, but we've stayed in the language of nucleic acids. Now, if we want to go to translation, well, that's not so easy to do, because in order to do translation, we need the English to Spanish, the Spanish to Chinese, the nucleic acid to amino acid dictionary. And that dictionary is the genetic code. So here is the genetic code, and this allows us to do the translation. Everything in black here is a codon, or a set of 3 bases, nucleotides, in the RNA. And everything in blue or red is our amino acids. So the translation of that. And you can see that we read these nucleic acids in codons in groups of 3. And so we basically use this table to find our sequence. So if I bounce back and forth, our first group of 3 nucleotides in the RNA is AUG. So if I come back to my table, well, the first letter is an A, so let me focus on this row. The second letter is a U, so now I'm down to this box, and then I can find that AUG is methionine. The second codon in our sequence is CCG. And so CCG is first letter C, let me focus on this row, second letter C, let me focus in this box, third letter G, that's proline. And then the third codon is UAG, and UAG is going to decode as U in the first letter, A in the second position, G in the third, and that happens to be a stop codon. Now that's why I was actually slowing down in my speaking while I was describing the genetic code, and I said I can't think and write at the same time. Sorry about that. I was actually trying to remember my stop codon, so I'd actually have a true stop here. Trying to remember that UAG was one of the stop codons. So if we go back to this, now we have done the translation. The amino acid chain that we're going to make here is methionine proline. There's nothing in the third position because stop just means stop. That means the end of translation. So this would encode a methionine proline dipeptide, two amino acids linked together. So that's the general idea of the central dogma. We go from DNA to RNA to protein. We can do this just from our head as long as we remember that thymines become uracils. We don't need any aid to do transcription, but translation will always require the genetic code. And it goes without saying, hopefully you all know me well enough by now. We've been together for 10 weeks or so. You'll have this on the third exam. You'll always have access for this on the exam and on the quizzes if you need it. Never is there going to be the expectation that you know this by heart. I don't know this by heart, and I've been doing this stuff for 30 years. So let's shift gears almost seamlessly and begin talking about the next lecture, because the next lecture is all about mutations. And what I'd like to call your attention to right off the bat is that there is redundancy in this genetic code. What we see is that multiple codons code for the same amino acid. In other words, if your DNA codon is TCA, that's going to ultimately call for a serine when you're undergoing translation. But if you happen to be TCC, that's also going to have a serine in there. So many different codons code for an identical amino acid, and that has important implications that we'll get to in just a little bit. But this is that cheat sheet that you're going to be getting through a Canvas announcement, either right before you watch this video or right after. It'll be released on Canvas right around the same point of this video. And you can see it's truly a study guide on these terms. So it'll be up to you to learn the definitions. It'll be up to you to know these definitions and be able to apply them. But I wanted to give you a one-stop shop of all of the most important terms. And we can use this video here to kind of review these terms. So I'll put this off to the side. And we'll go through these very quickly, but at least you'll have access to this short video, and they'll all be listed together, so you don't have to go back to the main video to sort out what's what. So somatic and germline get at location. This gives you the idea of the cellular location of the mutation. Germline mutations are in the gametes, and somatic mutations are in the body cells. Now what that means is if we have a somatic mutation, we only have that mutation in that one cell or collection of cells in the body. So the most common and probably the most relevant example of a somatic mutation is mutations or accumulated mutations that cause cancer. That's in our own body cells. Germline mutations don't impact the person who has that mutation. In other words, we don't use our gametes for ourselves. We only use our gametes for making the next generation. So a germline mutation within ourselves is not going to hurt us. But when we use that mutant gamete in a round of sexual reproduction, the only genetic information we're giving that next offspring is mutant information. So somatic mutations are localized into the one body region of the individual who has the mutation. Germline mutations don't affect the individual who has it at all, but they're passed into ultimately all of the cells of the next generation, because that next generation offspring only has that one genome to build off of. It only started from that single cell zygote, and that single cell zygote had that germline mutation in it, and so all subsequent cells has it as well. So somatic and germline, body cells, and gametes. Spontaneous mutations versus induced mutations give us an idea of cause, not location, but cause. Spontaneous mutations are due to cellular errors. They happen all on their own. And induced mutations are environmentally caused. These are induced or caused by external influences. So the most common cellular error that we talk about in this class are DNA replication errors, the mistakes that our enzymes make when they're copying our genome. Again, spontaneous. Nothing caused it. This was a cellular mistake. This was a cellular accident. As the DNA polymerase enzyme was copying our genome, it made an unforced error and introduced a mutation into those new chromosomes, into those new strands in semi-conservative replication. Induced mutations come from the outside environment. This can be tobacco smoke. This can be drinking contaminated water. Anything, X-rays, anything that is inducing a mutation in us from the external environment is going to be an induced mutation. So cause, not location. The next item on our list are double-strand breaks. Double-strand breaks, pretty self-explanatory. This is when we have a cut or a break in both strands of the double-stranded DNA molecule. So truly a double-stranded break. And there are major implications in the causes of these mutations, the results of these mutations, and how radically the cell goes about fixing these. We don't talk about that too much in this class, but we'll get a lot of that in cell and molecular biology. I will kind of deviate off of this paper just a second, though, and say that we touch on, just very, very slightly touch on, the two different repair mechanisms for fixing double-strand breaks, and they are homologous recombination, same kind of recombination that causes crossovers in meiosis, and non-homologous repair, or non-homologous end joining. Non-homologous end joining. Homologous recombination is a very, very complex action that, again, we go over in detail in Bio 320. But for this class, all we have to know is that we fill in the missing information of the break by stealing the corresponding information from the homologous chromosome. So we use the homologous chromosome from the other parent as a guide for what we can put in here. And that's all you need to know about homologous recombination for our class. Non-homologous end joining is a very, very quick and dirty repair mechanism. Essentially, the cell just grabs each of these broken ends and glues them back together to make a continuous chromosome molecule. The reason why that's a quick and dirty repair mechanism is because it usually results in the loss of genetic information. But again, that's something for us to explore in cell molecular. Now, we have chromosomal versus gene mutations. This is an easy one to review because we've already talked about so much of this already. I don't even have to draw anything. Chromosomal mutations are chromosomal rearrangements. These are the things that we've already covered in Unit 2. You've already been tested on it. So tandem duplications, displaced duplications, translocations, inversions, all of that stuff, those were chromosomal mutations. Gene mutations is what we're talking about here. In fact, everything else on this page is a gene mutation. So any mutation that affects a single gene rather than the entire chromosome is a gene mutation, and that's really what this lecture is focusing on. Transitions and transversions. Now, a transition is when we stay in our nucleotide class, and transversions is when we leave our nucleotide class. Well, what do we mean by nucleotide class? There are two classes of nucleotides. They are purines and pyrimidines. And the purines are the doubled-membered rings, so they're the bigger nucleotides, and the pyrimidines are the single-membered rings. They're the smaller ones. Now, first mnemonic, first memory device. Purine, small word, right? Big ring. Pyrimidine, big word, extra syllable, small ring. That's the way I remember those. So remember that they're opposite. The size of the word is opposite the size of the ring. The second mnemonic that I use is the made-up word purag. And what purag tells me is that adenine and guanine are the purines, which means adenine and guanine are the larger bases. If adenine and guanine are the purines, then cytosine and thymine, those are your pyrimidines. So if you remember a small word, big ring, big word, small ring, and you remember purag, you'll keep everything straight you need for this material. Keep in mind, A does not base pair with G. Our base pairs are always a purine with a pyrimidine. A base pairs with T. G base pairs with C. That isn't changing. It's just the nucleotide class they belong to is different. Adenine and guanine are the purines, and adenine base pairs with the pyrimidine T, and guanine base pairs with the pyrimidine C. So with the classes reviewed, we can make much more sense of transitions and transversions. If transitions stay in their class, then the only way that we can have a transition of adenine is to guanine. And the only way we can have a transition of cytosine is to thymine. And guanine can transition to adenine, and thymine can transition to cytosine. We stay in our class. In other words, a pyrimidine mutates to the other pyrimidine, or a purine mutates to the other purine. That's a transition. You've probably figured it out already, even if you don't remember it from the lecture. Leaving class is the opposite effect. If adenine becomes a thymine or a cytosine, we have a transversion. In other words, if a purine becomes either of the pyrimidines, we have a transversion. Likewise, if guanine becomes thymine or cytosine, we also have a transversion. Guanine is a purine, purag, and thymine and cytosine are the pyrimidines. If guanine becomes a thymine or a cytosine, we have a transversion. If thymine becomes an adenine or a guanine, we also have a transversion, because that's a pyrimidine becoming one of the purines. And if cytosine becomes an adenine or a guanine, we also have a transversion. So if a pyrimidine becomes a pyrimidine or a purine becomes a purine, we have a transition. And if a pyrimidine becomes a purine or a purine becomes a pyrimidine, we have a transversion. And that's what those mean. All of these, everything we've been talking about, is a base substitution. In other words, one base or nucleotide of the DNA has been substituted with another. And we can also call this synonymously a point mutation. A point mutation is when a single nucleotide is mutated. Just one nucleotide is mutated. That's a point mutation. So you can see we're beginning to layer terms. We're going to come back to this in a little bit. But all of these are base substitutions. All of these are point mutations. Transitions, you stay in your class. Transversions, you leave your class. Types of mutations. These are all types of mutations now. The last type of mutation is an indel. And an indel stands for insertion deletion. We're either removing bases or adding bases. Base substitutions is when one base becomes another. Indels are the removal or addition of a base. So if we go back to that sequence that we had earlier, I think this was the sequence I had down. I apologize if it's a little bit different. An indel would be something like this, where we've lost a C. An indel would also be something like this, where we've gained a T. So here in this sequence, this C was removed, and here in this sequence, this T was added. So we had a deletion or an insertion. That's an indel. Both of those are indels. Now, indels cause frame shifts. Remember we talked about the reading frame. Remember we were talking about codons earlier in this video. This is a methionine. This was a proline. This was stop. And that was a really nice clean reading frame. We just took these in groups of 3. Well, let's look and see what indels do. If we have a deletion, there's a methionine. Ooh, I'd have to go back to the genetic code to see what this is. And that's not even a stop codon, because we've only got 2 bases there. Who knows what comes next? We have shifted the reading frame. We've disrupted this decoding here. That's for the deletion. In the insertion, that's still methionine. And again, I'd have to go back and see if this is either proline or not. So we have a potential change there. Well, now we have a third codon, so we're going to add an amino acid because this isn't a stop codon, and we'll actually keep going down the DNA until we ultimately reach a new stop codon. So again, we have shifted the reading frame with this insertion. We've completely disrupted and scrambled the amino acid sequence. So indels are wicked mutations, and they lead to very dramatic frame shifts because we don't affect just one nucleotide, we don't affect just one codon, we actually shift and scramble the entire reading frame from the indel forward. So really major potential consequences there. So that's an indel, and it leads to a frame shift. Kind of talk about that in a pair. Finally, expanding nucleotide repeats. We won't talk too much about this in this class. This is really just for the forensics majors who are taking the class. The absolute basis, the entire foundation of DNA fingerprinting relies on expanding nucleotide repeats. So many of us have these islands, they're called, these regions on our chromosomes that are just junk DNA, and they are these nucleotide repeats, meaning a small set of nucleotides, in this case CTAG, that just repeats some n number of times. And it goes on and on and on, often for dozens and dozens of repeats. This is not a gene, so it's never going to be decoded. This is never transcribed or translated. Again, it's junk DNA. But for reasons that are very molecular, for reasons that we'll talk about in CellMolec, these types of DNA sequences expand or contract pretty easily. In other words, I, in my genome, might have 12 of these repeats, but it's not uncommon. It's actually quite easy for someone else to have 14 of them, for someone else to have 9 of them. Again, these very easily mutate to either expand and grow in number or contract. And since these aren't genes and they don't code for proteins, there's no ill effects of that kind of mutation because it's in junk DNA anyway. But it allows us to begin to identify one person from another. So if I committed the crime and you can identify that I have 12 repeats in the biological material that I left behind at the crime scene, and you have a suspect in prison, in custody, and you check their DNA and they have 14 repeats in that same location, that's not your perp. That's not the person who did it, because you already know that the perpetrator DNA has 12 repeats. So that's the basis of DNA fingerprinting. Again, all those are types of mutations. Now we move on to the effects of the mutations. And this is where things get interesting and we start to pull material together here from everything in this video, everything in the lecture. So we have silent mutations, missense mutations, and nonsense mutations. Let's focus on those three first. So a silent mutation has no effect at all on the protein that we're making. No effect whatsoever. A missense mutation changes one amino acid to another. So that is an effect. Different amino acids have different functions in a protein. If we change one amino acid and substitute it with another, the protein will likely have an altered function. Nonsense mutations is when we change the amino acid to a stop codon. That's the end result of the mutation. So in order to understand how these work, we actually need to go back to our genetic code and the redundancy of that genetic code. So let's imagine a codon in the DNA that's CTT, and we're going to mutate that. Let's get a little bit fancy and say that it's a transversion mutation. So a transversion mutation. Now we're starting to pull things together. So we want CTT, and we're going to have a transversion mutation in this third position. Well, what is a transversion mutation? It's a base substitution or a point mutation. We're only affecting one base. So good, that checks out. And we're not going to remove or add to that base. That would be an indel. We're just going to swap that base for another, base substitution. So the question becomes, what's that thymine going to become? Is it going to become an adenine, a cytosine, or a guanine? Well, what did I say? I said it was a transition, I hope. That's what I meant to say. So if it's a transition, we have to remember that that means we stay in our class. So pure ag was one class, and CT was the other class, the pyrimidine class. And if it's a transition, then we're going to stay in our class. So CTT is going to become CTC. This thymine, which is a pyrimidine, is going to become a cytosine, which is also a pyrimidine. That's our transition. It's going to be a transition mutation. Okay, so we've categorized its type. Now we need to think about, well, where is this mutation? If it's in my skin cell, how else can I classify it? Well, if it's in my skin cells are not gametes, so that would be a somatic mutation, right? It wouldn't be a germline mutation. So that term applies here as well. Now, in order to get to effect, in order to get to these terms, we have to dig a little bit deeper into the molecular genetics of it. We have to begin thinking about the decoding of this. So first, let's do transcription. I don't need a table for transcription. This codon would transcribe to being that, and this codon would transcribe to being that. And now, if we're going to do translation, let's go to our genetic code for translation, and CUU codes for, where are you? CUU codes for leucine, where CUC codes for, what does CUC code for? Well, it codes for leucine. So even though we had this base substitution, somatic mutation, transition mutation of a thymine for a cytosine, because of the redundancy of the genetic code, we actually have a silent mutation. There's no effect of this mutation because a leucine is still a leucine in the protein, and a protein is all that matters. Genes exist to make proteins. Proteins keep the cell alive. If the protein is unchanged, the mutation is silent. It has no effect at all. So, great. Let's go a little bit further here. Let's go with another codon. And now, let's say that in this codon, we are going to mutate a, how do I want to do this? I'm just thinking out loud here. We want to mutate a CAT codon, and this time we're going to have a transversion. Let's make it a transversion this time. So we'll go back to the board. We have CAT in the DNA, and it's going to be a transversion. So point substitution, right? Definitely not a chromosomal rearrangement, not a chromosomal mutation. So this is a base substitution. And if this was happening in a spermatocyte, it's going to be germline. That tells us where in the body it is. But the pertinent information here is that it's a transversion. So if it's a transversion, I have to give you a little bit more information because that means it's a pyrimidine becoming a purine. And the next question becomes, well, which purine is it becoming? Well, let's say it's becoming a guanine. So that makes it a transversion. We've gone from pyrimidine to purine. And then we want to go through transcription. Excuse me. Maybe I'll edit that out. Maybe I won't. If we go through transcription, we're going to do a CAU. Remember, we're just changing the thymine to a uracil. Transcription here doesn't have to change anything because this is CAG. And then for translation, we got to go back to our genetic code now, don't we? So if we go back to our genetic code, we see that CAU, C to the A to the U, encodes histidine. And you don't have to know what these three-letter codes code for in this class. You'll get that in biochemistry. So if you see his, just put his. And CAG codes for glutamine, GLN. So let's say this isn't a silent mutation because the protein was changed. One amino acid has been substituted by another. That's a missense mutation. That's what we mean by missense. Now, what about a nonsense mutation? What happens in a nonsense mutation? Well, in a nonsense mutation, what we have is, let's pick up with that CAG, and let's have a transition in the first position. So if we go back to the board, we see we have CAG. That's a point mutation, right? Base substitution. We know that. Whether it's somatic or germline, we would need more information. Let's say that this is in a liver. So if I tell you this mutation occurred in a liver cell, you would know it's somatic, not germline. And I said a transition in the first position. So there's the first position. That's a pyrimidine. So we have to change to the other pyrimidine if it's a transition. And that's the effect of the mutation. Let's go through transcription first. That would be transcription. And now we go to translation. So before the mutation, CAG codes for CAG, right? We did that already. That's glutamine. So that was before the mutation. That's the wild type sequence. And then after this transition mutation, we became a UAG codon. Well, UAG, that codes for stop. Ooh, not good. We've truncated the protein. There's going to be no further translation after this mutant codon. Even though there are codons after this mutant codon that are supposed to be part of the gene, supposed to be part of the protein, even though this gene keeps going, it's not going to happen. Translation won't occur because once this stop codon is reached, translation halts. So when one amino acid is mutated and the codon that codes for that amino acid becomes a stop codon, that's your nonsense mutation. That's what we mean by a nonsense mutation. So I would like you to begin getting comfortable with these terms in the kind of multi-layered way that I'm trying to emphasize to you here. If I set you up with a specific example and I tell you that this mutation, let's say it's this mutation. If I tell you that in an oocyte, a CAG codon mutated to a TAG codon, please characterize that mutation using as many terms as possible. That's a lot of information. So oocyte, that's a gamete. So you know that's going to be a germline mutation. I didn't tell you anything about what caused it, so if it was a cellular error, it would be spontaneous. If it was caused by radiation exposure, it would be induced. We know it's not a double-strand break. It's definitely a gene mutation. One base is becoming another, so it's a base substitution point mutation. The mutation has a pyrimidine going to a pyrimidine, so you can call it a transition. And once you go through the decoding, you realize that it's also a nonsense mutation. So many of these terms apply to single mutations, and we want you comfortable doing that and categorizing these mutations in that way. That's why we're spending so much time reviewing it here. The last pair of terms that are on here before we get to secondary mutations is loss of function and gain of function. So loss of function and gain of function. Loss of function and gain of function mutations get at protein action. So when there is a loss of function mutation, the protein that's ultimately made has less or no function compared to its wild type. So the mutant protein has less or no function. And gain of function mutations is when the mutant protein that's ultimately translated from the mutant gene has more or higher function. It does more. Sometimes it's unregulatable, so that protein can't be turned off. It's just always active. Sometimes it's gained new functions and does more things than it's supposed to. So loss of function mutations, the mutant protein has less function or no function. Gain of function mutations, the mutant has more activity. Now, this has implications for Mendelian genetics. Imagine we have the wild-type unmutated protein encoded by the wild-type unmutated allele. But we're all diploid. We have two alleles, one from each parent. And imagine that the other allele is the mutant allele. So we've got a wild-type allele and a mutant allele. For the loss of function mutation, when there's a mutant protein that's attenuated, it's weaker, which of these two proteins do you think is stronger? The one with full function or the one with less or no function? That's right. This allele is stronger because it has more function than this. This is the stronger allele, and stronger alleles are dominant. So loss of function mutations are recessive mutations. I love it. Now for this scenario, which of these two proteins do you think is stronger? The one with normal function or the one with more or higher function? Well, in this case, it's the mutant protein that's stronger because the mutant protein has even more function than the wild type. In gain of function mutations, the mutant allele is dominant. Gain of function mutations are more rare, but they're out there, And this is how we can get dominant mutant alleles. So really, really nice tie-in between molecular genetics and Mendelian genetics right there in the context of mutations. Finally, wrapping up with secondary mutations, we have reversion mutations and we have suppressor mutations. Reversion mutations are easy to wrap your head around. So let's start with an example codon, TCT. So TCT is obviously a codon, right? It's going to transcribe to UCU, and it's going to translate to what? It's going to translate to UCU, translates to serine. And again, you'll always have the genetic code. And now let's pick up a mutation. Let's say that it's going to be a transversion mutation in the first position, and let's transvert over to an adenine. So the mutant base substitution here is going to be an adenine mutation, base substitution, transversion. Pyrimidine became purine, and that will transcribe to ACU, and ACU is going to be, where is it? ACU, threonine. So we can apply our terminology to this, right? So this is a base substitution point mutation. One base became another. It's a transversion. A pyrimidine became a purine. And it's a missense mutation. A serine became a threonine. One amino acid became another. Now, if we're going to do a reversion mutation, the reversion mutation here would be a second mutation that mutates this adenine back to a threonine. Just move suppressor down here. We'll deal with that in a second. So reversion is a true reversal. It was supposed to be a threonine. It got hit with a mutation. Let's say this mutation was due to a replication error, so that means it would be spontaneous. The spontaneous mutation led to a transversion missense mutation. Serine became threonine. This is a mutant protein. And now let's say that very same nucleotide got hit with ionizing radiation that induced a mutation. So this is an induced mutation, and that adenine became a thymine again. Well, we already know that that thymine is going to transcribe, well, that whole codon is going to transcribe as UCU, which is serine. So we've gone back to the original amino acid. We've restored the wild-type sequence of the protein. That's a reversion mutation. We mutate from one base to another, and then the secondary mutation mutates us back to the base we were supposed to be. For a suppressor mutation, let's use the same initial mutation. It's spontaneous because it was due to a DNA replication error. It's a base substitution point mutation because one base is turned into another. It's a transversion because a pyrimidine became a purine. And it's missense because one amino acid became another. We mutate to a threonine. It's a pretty dramatic shift. It's going to have a bad effect on the protein. But now let's say we're going to get a secondary mutation, but the secondary mutation is not going to affect the first position. It's going to affect the second. So we're going to keep the mutant A, right? That was our first mutation. But now we're also going to have a transversion of this cytosine to a guanine. So our initial codon was TCT, which transcribed to UCU. Then we had our first mutation where the T became an adenine, and that was our missense mutation. The secondary mutation alters the codon even more, which transcribes to AGU. The question becomes, what does AGU code for? Well, AGU, AGU codes for serine. So we actually do go back to the initial amino acid. We wanted a serine there in the wild-type sequence. We still have a serine there after the secondary mutation, but this isn't a reversion mutation because we didn't go back to the original codon. This is a suppressor mutation. So a suppressor mutation is a mutation that is different than the first. It affects a completely different region than the first, but it still returns us to the wild-type protein sequence, and at the end of the day, it's the protein sequence that matters. All right, so to wrap up here, I'm just going to go back to my notes because I think there was one more thing that I wanted to cover. We talked about the 5' and 3' ends, conservative replication. We went over the central dogma, the redundancy of the genetic code. We went through all of the mutations, yay for us, so a nice little resource for you to have these all in one place. and it's just the Ames test. So the Ames test is a carcinogenicity test. We use the Ames test to see if chemicals that are being used in products around the world can potentially cause cancer. Cancer is a disease of accumulated gene mutations. So essentially anything that's mutagenic to DNA, any compound that would cause mutations in DNA can cause cancer. So by screening for a molecule's ability to cause mutations, we're actually screening for that molecule's ability to cause cancer. So the Ames test starts off with already mutated bacteria. And those bacterial cells, they're grown in a culture. Since they're already mutated, they can't make an amino acid they need to survive. We can use the example histidine. I'm not sure. I don't remember if that's what I used in the lecture or not, but it's common for the Ames test. So we start with bacteria that can't make their own histidine. Now, of course, we can keep these bacteria growing because if we add histidine to their media, if we add histidine to the food that they're growing in, they'll survive. These bacterial cells need histidine to make their proteins. They need histidine to live, but they don't really care where they get it. If they can't make it on their own, but they're getting it from their environment, they happily live. So these are cells that can't make their own histidine, but can grow when we supplement histidine to them. And the reason why they can't grow their own histidine is because of a base substitution mutation in their genome. A single base is mutated. And then what we do is we take some of those cells, and to one batch of those cells, we add the compound we're studying. And the other batch is a control. It gets no compound. And we take those cells after they've grown a while, either in their control or their compound environment, and we grow them on food, on media, on plates that lacks histidine. So now these cells are not getting histidine from their environment, and they're not getting histidine from themselves, because they shouldn't be able to make it. We expect to see maybe 1 or 2 colonies on this plate. These are spontaneous mutations that were either reversion mutations or suppressor mutations that fixed the mutation here, allowing the cells to become His+. But it's very, very infrequent and very, very uncommon. If a compound causes mutagenesis, we will see a lot more colonies on this plate because this compound increased the mutation rate. And by increasing the mutation rate, it increased the suppressor and reversion mutation rate, so a lot of cells became His+ and were able to grow in the absence of histidine. That would tell us that this compound promotes DNA mutations, and anything that promotes DNA mutations promotes cancer. Conversely, if we grow the cells in the presence of our compound, and only 1 or 2 colonies grow, well, that was the point of the control. The control shows us that that is the background rate of suppressor and reversion mutations, even when there's no carcinogenicity around. So this would be a negative result, and the compound would be determined to be safe for use. So a really, really nice and simple test, no harm to animals, just uses bacteria. We start with a mutated cell, but what are we going after? What are we really screening for? We're really screening for reversion and suppressor mutations in the Ames test, but an increase in reversion and suppressor mutation, as evidenced by more cells growing on minus histidine media, is an indicator of overall increase in mutation rate. That's an indicator of carcinogenicity. All right, so a pretty long one, but hopefully a useful one all the same. A little bit clearer on the terminology, I hope. And we'll see how you do on the challenge questions. As always, thanks for watching. And if there are any remaining questions on this material or anything else from the week so far, you know you can put those questions to me right here.